Hello, I'm Jonathan Barratt from the University of Leicester in the UK, and I'd like to thank the organisers for asking me to give a high-level overview of the HIF stabiliser trials in CKD and dialysis-dependent patients. What I'm going to try and achieve over the next 20 minutes or so is give you an overview of the clinical trial landscape, the key results from those clinical trials, and the major unanswered questions that the data give us and which we are trying to grapple with over this Controversies conference. And I hope this sets us up for an interesting discussion at the end. I'm sorry I can't be with you directly for this talk, but I should be able to join for the questions later. So I'm going to turn my video off now and to not distract from the slides and move on. Here are my disclosures. And I just want to mention that I have received speaker fees and attended advisory boards for both Astellas and GSK that are relevant to this presentation. As I've already mentioned, I'm going to give a very high-level overview of the HIF stabiliser trials, and I'm going to actually focus on the devil in the detail, because this is where the challenges arise for us at this Controversies meeting, and particularly within the work groups, when we try and compare data across studies within a single programme and across programmes of studies by different sponsors. And I'm sure many of the things I'm going to mention have already been rehearsed and discussions begun within the work groups. I'm only going to focus on HIF stabiliser trials that have been performed globally, and essentially what that means is it is limited to the data available for daprodustat, roxadustat and vadadustat. I accept there are other HIF stabilisers in development, and there's a lot of significant trial work that has come out of Japan, which has focused on Japanese patients with non-dialysis dependent and dialysis dependent CKD, but I'm not going to discuss those. I'm going to limit this discussion simply because of time to those studies that are being evaluated in global trials. And so when we consider these studies, we have daprodustat, roxadustat and vadadustat, and as you can see here, all evaluated in global programs. At the moment, daprodustat is approved for use in Japan, and there are US and EU filing planned. Roxadustat has been launched in Chile, Japan, China, South Korea, Europe and the United Kingdom, and there is a filing in process in the US. And vadadustat has been approved in Japan with US and EU filing planned. You can see here some of the high-level differences between the drugs in terms of half-life and in terms of expected dosing, with daprodustat likely to be given as an oral once-daily medication, as would vadadustat, whereas roxadustat, having a longer half-life, is given orally 3 times per week. Here you can see summarised the global trial programs for daprodustat, roxadustat and vadadustat. As you can see, there are some differences in the patient population studied, and I think this is important, and I'll touch on this in a minute, that we are looking at separately non-dialysis dependent CKD patients, we are looking at incident dialysis dependent CKD patients, and patients who are stable on dialysis. We are looking at patient populations who are ESA naive, or patients that are already maintained on ESA and converted to a HIF stabiliser. We need to look again at the comparator used, and some studies have been placebo controlled, not many, but most have involved an active comparator. And that active comparator has either been darbepoetin alpha or other recombinant human EPOs such as epoetin alpha. For the ASCEND program evaluating daprodustat, you can see here a little bit more granularity. You can see the studies in non-dialysis dependent CKD are the ASCEND NHQ study, which is a placebo-controlled study in ESA naive patients, the ASCEND non-dialysis study, which included both those ESA naive and ESA treated patients. You then have an incident dialysis dependent CKD study, ASCEND ID, and you have the stable dialysis dependent CKD populations, ASCEND D and ASCEND TD. I'm not going to discuss those studies in grey. I'm really going to focus on those studies that are published phase 3 studies in the daprodustat program. In the Alpine program evaluating roxadustat, you can see here in the non-dialysis dependent population, you have placebo-controlled studies. in ESA-naive patients, the ALPS, the Olympus and the Andes studies. And you also have the Dolomite study, which was comparing roxadustat to darbepoetin alpha in ESA-naive, non-dialysis dependent patients. In the incident dialysis dependent CKD population, you have Himalayas, which was exclusively ESA-naive patients. And then you have the Rockies and Sierras, which you can see straddle both incident dialysis dependent and stable dialysis dependent patients, with the Rockies including both ESA-naive and ESA-treated, and Sierras exclusively evaluating ESA-treated patients. And then you have the Pyrenees study, which was performed in stable dialysis dependent CKD patients, all of who were previously treated with erythropoietins. And finally, we have the vadadustat program, the PROTECT and INNOVATE trials. And PROTECT was a study in non-dialysis dependent CKD patients, including both ESA-naive and ESA-treated patients. And the INNOVATE study was a study in dialysis dependent patients with subgroups both within the incident population and within the stable dialysis dependent population. Programs, and I'm sure I'm not saying anything that a lot of you have thought about previously and have been commented on, and I'm sure these are hot topics for discussions within the work groups. And the disclaimer here is that I offer no solutions or answers to these, but I want to raise them because I think they are important whenever we are looking at the data that I'm going to present next. And the first thing to say is that these global programs have differences in the relative contribution of different regions, and this contribution is different across programs, but also within studies within programs. And this may well influence our interpretability of the results. So patient phenotype by region may differ in terms of ethnicity, starting hemoglobins, comorbidities and concomitant medications, and all of these may influence the outcome both in terms of efficacy and safety of HIF stabilizer use. And we've already seen in the literature some comments regarding this, and I think this is really important when we start looking at comparing studies within programs and across programs. In terms of the analytical approaches to assess efficacy and safety, again, when we compare across programs and even studies within the same program, different approaches have been taken in terms of non-inferiority versus superiority, in terms of the outcomes that are measured and the hierarchy used. Sensitivity analyses vary between studies and across programs, and we need to question, and I know we're going to have a specific plenary session on non-inferiority studies, but I think the reliability of a non-inferiority in small subgroups needs to be questioned as to whether that truly shows non-inferiority or whether it's able to show non-inferiority. And this is where I think we might need to think about combining data across programs to be able to tease this information out in the long term. When we think about efficacy and safety profiles, and here I'm talking about cardiovascular and thromboembolic risk, this is likely to vary dependent upon the patient population. Basal risk is going to be different across non-dialysis dependent, incident dialysis dependent, and stable dialysis dependent populations, as we all know. And we need to take this into account along with potentially the impact of prior ESA exposure. Is this important? Does this prime for potential safety signals when this is followed by a HIF stabilizer? We don't know that at the moment. And of course, does the ESA comparator matter? There are different ESA comparators across studies. Do we know that one ESA is likely to have the same impact as another? Again, areas that we need to really think about and discuss when we're comparing data across studies. And finally, I know there's a lot of talk about whether we're seeing class effects or whether there are drug-specific effects. There is exciting preclinical data looking at the different mechanisms of action of the different HIF stabilizers. Can the data across the clinical trials that we have available to us answer the question as to what is truly a class effect and what is truly a drug-specific effect? And that comes down to the devil in the detail in terms of how these studies were delivered and can we compare directly across studies? And that comes down both to efficacy and safety. And I'm going to try and draw out some of those differences for the remainder of my talk. So if we start by looking at the non-dialysis dependent CKD population of the placebo-controlled trials, I'm going to present a series of tables for different patient populations. It's going to contain a lot of data which I'm not going to go through. This is high-level data. Really, I'm using to tease out some important points. So here we have the Ascend NHQ study for daprodustat and the roxadustat ALPS, Olympus and Andes studies. And you can see here the numbers of patients. Both were dealing with the hemoglobin correction phase. Treatment periods, evaluation periods were different. Primary endpoints were different here and different between the US and the EU. Target hemoglobins were different. Different approaches to analysis have been adopted. And there were additional key endpoints were different between the studies. And so this brings me on to my first set of challenges, which are common across all of the studies, you'll see. The first is how important is the difference in treatment period and HIF stabilizer exposure? How important is the evaluation period and the target hemoglobin? The definition of primary outcome and the reported secondary outcomes and their definitions. We have an important difference here in terms of the approach taken in studies conducted in the United States against those studies conducted outside of the United States in terms of target hemoglobin and definitions of primary outcomes. And this does make looking at the data more challenging, and particularly with regard to factors we believe may be important in safety in terms of cardiovascular events, different target hemoglobins, and how is that going to influence the data that we are looking at? As I say, I don't have any obvious answers for you, but I think these are things that we clearly need to be considering when we are trying to tease out data across studies. If we now look at the ESA-controlled studies in the non-dialysis dependent CKD population, you can see we've got the Ascend ND, Dolomite, and the Protect studies. You can see the comparator is the same, darbepoetin alpha, but actually these studies are looking differently at the different phases of renal anemia correction. So we've got hemoglobin correction and ESA conversion for daprodustat and vadadustat, but only hemoglobin correction with roxadustat. You have different treatment periods, different evaluation periods, primary endpoints assessed at different time points, again different target hemoglobins, and a different set of additional key endpoints, secondary endpoints that were evaluated. And so now when you look at my list of challenges that we face when interpreting the data, you'll see added to this list is the impact of correction versus conversion. Do we know that the risk profile associated with hemoglobin correction is the same as converting a patient from a stable dose of ESA to a HIF stabilizer? Can we be sure that the conversion factor used is appropriate? Has it been the same across studies? And it hasn't been. The process of converting has not been identical across studies. What is the impact of the magnitude of correction and starting hemoglobin? What is the impact of a delta change in hemoglobin? And importantly, what is the impact of a rate of change of correction of hemoglobin? All of these factors are potentially important in the safety outcomes associated with these studies. And we really need to try and tease this information out if we're going to compare across agents. Before we assume that one drug is behaving differently to the other, we need to be sure that we're dealing with a stable background and a stable process for which these drugs have been used. And that's not the case in most of the studies that we have available to us at the moment. If we now move on to the incident dialysis dependent CKD studies, you can see here Ascend ID, Himalayas, Rockies and Sierras for roxadustat and Innovate for vadadustat. You can see the comparator is different across the studies, and again the studies are variably looking at hemoglobin correction or hemoglobin correction and ESA conversion. The treatment periods and evaluation periods again are different. The primary endpoints again are different, as are the target hemoglobins and the additional key secondary endpoints. And so again, this is bringing in more heterogeneity when we look in this particular population. And now you will see added to my list the pros and cons of pooled analyses. There are certainly advantages to pooling data, But actually, what do we lose when we pool data? And I would imagine that's going to be a key discussion point during this conference, because there are a number of publications looking at pooled analyses, and we need to think about how reliable they are and at what cost does pooling data come with. We also need to start thinking about how we view the iron data within these studies, both the baseline parameters, but also the usage of iron, which has been a key area of interest across studies. Jodie is going to follow this talk specifically looking at iron usage, and I'm not going to go into that in great detail, but I will comment a little bit on that later in the talk. And finally, we have the ESA-controlled studies in the stable dialysis-dependent population. Here we have Ascend-D, the Pyrenees, Rockies and Sierras studies in roxadustat, and the Innovate prevalence study with vadadustat. You can see the comparators were different across the study programs. You can see that actually all were dealing with ESA conversion, but interestingly for daprodustat, they included ESA responders and non-responders. The roxadustat studies looked at ESA responders only. Treatment periods again were different, as were evaluation periods and primary endpoints. Target hemoglobins again different. Again, there's been pooled analyses performed, and the key additional endpoints are different across studies. And so now added to my list of challenges are, how do we handle ESA responders compared to non-responders? What does this tell us about the action, the efficacy and the safety of the HIF stabilizers? What are these patient characteristics? And should we be looking at these separately or not? And if they haven't been looked at separately, does that lead us to have less confidence in the data? The other factor that I just want to mention is dialysis vintage when we're thinking about stable dialysis populations, because of course we know the time spent on dialysis has a significant influence on morbidity and mortality, and may well impact on a number of outcomes measured in these studies. And so if we now look at the key efficacy outcome for the non-dialysis dependent CKD studies in terms of hemoglobin response, you can see that for each of the HIF stabilizers, non-inferiority was demonstrated to darbepoetin alpha in terms of the hemoglobin response from primary outcome. But you must of course remember that the target hemoglobins were different. This dealt with both hemoglobin correction and conversion differently across studies. And the time to measurement of the response was different across these studies. But non-inferiority was demonstrated with each of the HIF stabilizers in this population. And here you can see that data presented graphically. If we now look at the hemoglobin response in the incident dialysis-dependent CKD population, you can see that for vadadustat in the Innovate study, non-inferiority to darbepoetin alpha was demonstrated. But note, this is a pooled analysis, pooling both the incident and prevalent populations. In the roxadustat study, the data has been presented as a pooled analysis of the Himalayas, Rockies and Sierras studies, and you can see that data here. And the Ascend-ID study of daprodustat has been presented but not published, and the data from that presentation is here. Of course, note that, again, the target hemoglobins were different. The time to measure the response in hemoglobins were different. And again, we're dealing with a mix of hemoglobin correction and ESA conversion within these studies. And here you can see that data presented graphically. And finally, if we look at the hemoglobin response in the stable dialysis-dependent CKD population, you can see that non-inferiority was demonstrated in the Ascend-D study of daprodustat against darbepoetin alpha. You can see non-inferiority has been demonstrated with vadadustat in the Innovate study, but remember this is a pooled analysis with incident and prevalent dialysis-dependent patients. And there has been a pooled analysis performed of the Pyrenees, Rockies and Sierras studies, and you see the data there. Again, just to remind you, different target hemoglobins and different timelines upon which the primary endpoint was measured. And here is that data presented graphically. A key area within all of the efficacy analyses has been the use of intravenous iron. I've put some summary data here in this slide in terms of the different populations. I'm not going to go into much detail because Jodie is going to cover this far more extensively in the talk that follows mine. But what I do want to do is draw out some important areas that we need to think about. And so you can see my list of challenges is now extended to thinking specifically about the iron data and really looking at how across these studies iron repletion and deficiency were defined, and that's different depending on the study. What the protocolized use of iron supplementation was within the program, within the study, or even within the arms of an individual study, because this has varied across the different programs. And what the target iron parameters were. This is going to be discussed extensively in the next talk, but I think these are really important high-level areas we do need to consider when we try and interpret the iron data. Another key area that we really are very interested in is quality of life data. And again, I've summarized some of that data here in this table, again, really just to draw out some points. And these are, do we know what the right tool is to measure patient-reported outcomes in either the correction phase of anemia treatment or during the conversion phase? Do we only need one tool? When is the right time to assess quality of life? Because it varies in the different studies. And should we use the same tool for the non-dialysis dependent and dialysis dependent patients where quality of life is clearly very different depending on what stage in the disease you are in? Aren't questions I don't have the answer to, but I'm sure these are things that we've been discussing in our work groups. And finally, the top-level safety data across the programs, principally looking at cardiovascular and thromboembolic safety signals. And here you've got the data from the roxadustat studies, placebo-controlled studies in non-dialysis dependent CKD patients. And you see both an on-treatment analysis and an intention-to-treat analysis for these endpoints of both MACE, MACE plus, and all-cause mortality. And so when we think about the safety analyses, which is right? Should we be doing intention to treat or should we be doing on-treatment analyses? And I think this is important because different programs have taken different approaches and we need a discussion about this in terms of the different programs and what that tells us about the safety signals and which is more appropriate in which setting. Here we have data for the ESA-controlled non-dialysis dependent CKD studies. What you can see here is that for daprodustat, the ASCEND-ND study, non-inferiority against darbepoetin alfa was shown for MACE. You can see that the non-inferiority margin was not met for vadadustat in the PROTECT study compared to darbepoetin alfa. And you can see other selected data on MACE and other cardiovascular endpoints in this table. But I just want to use this table really to bring out a couple of points. And these are, should we be thinking about non-inferiority for safety or should we actually be thinking about superiority? And this will come down to your position on whether hemoglobin correction with an ESA is safe and therefore non-inferiority is appropriate, or whether actually hemoglobin correction with an ESA is potentially unsafe and therefore we need to be showing superiority. And the other thing I just wanted to bring up is there have been a variety of different measures of safety employed. All have used MACE, but then we have additional measures such as MACE Plus and expanded MACE among a number of others, and which are the right ways to assess cardiovascular safety. Here we have the same data for the incident dialysis dependent CKD population, and again you can see presented both the MACE and other safety endpoints. For the vadadustat INNOVATE study, non-inferiority against darbepoetin alfa was shown for MACE, but of course you must remember that this is a pooled analysis between both incident and prevalent dialysis patients. Here is the same data for stable dialysis dependent CKD patients, again showing non-inferiority in the pooled dataset of incident and prevalent patients for vadadustat, and non-inferiority for daprodustat against darbepoetin alfa with regards to MACE in the ASCEND-D study. And finally, we have thromboembolic events. I've tried to summarize the event rates in this table, but what is clear is that the event rate was in fact low and the actual number of events was low across the studies, and this may be a situation where actually combining data sets may be useful to determine the true risk of thromboembolic events and whether there is any specificity to these events in relation to a particular agent. And so, as you can see, we've got a number of challenges to grapple with when we review the data available to us on HIF stabilizers in the treatment of CKD anemia. And I hope by Sunday we will have had a chance to deal with some of these, address and resolve some of the outstanding issues related to the use of HIF stabilizers in the treatment of renal anemia. Thank you very much for your attention.